This chapter discusses how chromosomes determine inheritance. And you can look at a couple Amoeba Sisters video about chromosomes and karyotypes, which is similar to what we did in lab, and look at sex-linked traits and how to do Punnett squares of sex-linked traits. And so we'll focus on how chromosomes determine genetic inheritance. So a few pre-chapter quiz questions to see where you're at. The relationship between genes and chromosomes is okay, so the correct answer is that genes are segments located on the chromosomes. So in humans, we have 22 pairs of autosomes and 1 pair of sex chromosomes. So 46 chromosomes altogether. And the smaller chromosomes have a few hundred genes along them. The larger chromosomes have a few thousand genes. So altogether, we have about 20,000 genes in our genome, about 20,000 genes in every one of our cells. Okay. Having an extra chromosome, a trisomy, arises from a non-disjunction, failure of chromosomes to separate in. Okay, so the correct answer is non-disjunction events leading to trisomies are possible in both meiosis 1 and meiosis 2. Apparently, people who have studied it say meiosis 1 is more common. It's more common that homologous pairs fail to separate rather than sister chromatids. But certainly, meiosis 2 non-disjunctions occur as well. And the normal human karyotype. Okay, so we have 46 chromosomes, and they're found in pairs, and the normal karyotype has 22 pairs of autosomes, non-sex chromosomes, and 1 pair of sex chromosomes. Okay. So scientists rediscovered Mendel's work from the 1850s in about 1900. He was one of these guys that nobody really understood his work in his day, and later on people rediscovered it. And by then, microscopes were better and people had an idea of the things called chromosomes, and they said, well, gee, it's not just particles he's looking at chromosomes and how chromosomes are passed on. So what are chromosomes? Chromosomes are thread-like structures built of DNA, and the DNA is wrapped around protein spools for organization. Just like in your sewing kit, the thread is what sews, but it has to be wrapped around spools to organize it. So the idea of chromosomes being what were inherited were developed by Sutton, who worked with grasshoppers, and Bovary, who worked with sea urchins. So animal models are often used to make discoveries that apply to humans. So it should be obvious to you now that you've done meiosis that the chromosomal theory of inheritance explains Mendelian genetics, and that during meiosis, chromosome pairs separate, and then gametes each contain only half of the chromosome complement, and then the gametes come together in fertilization so that each parent contributes equal amounts of chromosomes to the offspring. So this is what it says, that Bovary and Sutton worked with sea urchins, that was Bovary, and grasshoppers, that was Sutton, and showed that chromosomes occur in matched pairs that separate meiosis. And then a scientist by the name of T.H. Morgan discovered the X and Y chromosomes and which chromosomes are involved in sex. And he did it in fruit flies. So the little flies you see hovering around bananas sometimes. So that was also in the early 1900s. So by the early 1900s, we had a chromosome theory of inheritance. Here's a karyotype from humans, and here's a karyotype from a fruit fly. All right, so what determines if a human baby is male or female? It's not a single gene, it's not a few genes, it's a whole chromosome. And so here's an electron micrograph of the X chromosome and the Y chromosome. You can see that the X chromosome is much larger. It has over 1,000 genes. They don't all code for sex, they code for other things as well. The Y chromosome is a little shrimp, and the normal karyotype in a male is XY, so that's the male pattern, and the normal karyotype in a female is XX. She has 2 copies of the X chromosome. So men would have an XY, and when they undergo meiosis, the X and Y separate, so every sperm has 22 autosomes in an X or 22 autosomes in a Y. Every egg always has an X. Which of the 2 X's is random? So it's not always the same X, but an egg always has an X. So people will tell you that the man determines the sex of the child. It's an X and a Y when the baby's a boy. If it's an X and an X, then the baby's a girl. When homologous pairs pair up during meiosis, the X and Y have ends that match each other. So even though the Y is a little shrimp, it will find the X and match with the X and then separate. There's one gene on the Y chromosome, and we think it's pretty important in making men males. And it's a regulatory gene. We don't really talk about regulatory genes in this course, but it's a gene that turns on and off other genes. So its product regulates how other genes are turned on and off. So people sometimes say, well, a regulatory gene is like an orchestra conductor. So they're not playing an instrument, but they're directing which instruments to play. Okay. If a gene is located on either the X or Y chromosome, it's called a sex-linked genes. And especially with the X chromosome, there are a lot of genes on it besides sex. Now let's talk about red-green color blindness, a sex-linked recessive disorder. And that should explain how chromosomal inheritance works. So red-green color blindness is more common in men than women by far. People who have it have a hard time distinguishing red from green. About 1 in 12 men are colorblind, but only about 1 in 200 women. So color blindness is a recessive disorder, and it's found on the X chromosome. So the question is, why is it so much more common in men? Okay, so men have 1 copy of the X chromosome and then a Y. So if they have a faulty copy of that gene on the X chromosome, they have the disorder. Women have 2 copies of the X chromosome. So if they have 1 faulty copy, the good copy covers it up because it's a recessive allele. So they need 2 faulty copies of the gene in order to have the disorder. Okay? So if a woman has 1 faulty copy, they have normal vision, but they carry the disease. Okay, so for a color blindness problem, we show that it's on the X chromosome, and we do a superscript. So X to the capital N is having the normal color vision allele, and X to the little n is having the color blindness allele. So if you're a male, there are 2 possible genotypes. Either you're X to the big N with a Y, or you're X to the little n with a Y. If you have the normal allele, you have normal vision. If you have the colorblind allele, you have colorblind. Pretty straightforward. Okay, with a woman, it looks like any diploid gene. So if you have 2 copies of the normal allele, you have normal vision. If you have 1 copy of the normal allele and 1 copy of the faulty allele, you have normal vision, but you're heterozygous, and so you're called a carrier. You carry the disease, but you don't show it. Okay? And could a woman be colorblind? Yes, but she'd have to get it from both parents. So she'd have to have 2 copies and that's just like any recessive disorder, and then she's colorblind. Okay, so let's see how we solve a problem for colorblindness, and I'm sure I'll give you one of these on the exam. So a woman with normal vision, not a carrier, mates with a colorblind man. Okay, so for the man, I'll just tell you whether the man is colorblind or not. But for the woman, if she's not colorblind, I'll tell you whether she's a carrier or not. Okay? Predict the phenotypes of the sons, and for the daughters, also tell if they are carriers. Okay? So for the sons, well, let's just do it. The woman has normal vision, not a carrier, so she's X to the big N, X to the big N. The man is colorblind, so X to the little n, and then a Y. Okay, separate out the two chromosomes in meiosis. So every sperm would be either an X to the little n or a Y. Every egg would be an X to the big N because that's what she has. Bring them back together in fertilization. X to the big N across, X to the little n down. X to the big N across, Y down. X to the big N across, X to the little n down. X to the big N across, Y down. And we drew a little tail to show you this represents a sperm, that the X and Y are the sperm from the male, and the X to the big N are the eggs. Okay, then you just read them off, and you should read off the men and the women separately. Okay, so if they have boys, that means the man gave them a Y, and they got the normal allele from the mom. If the boys, they're all normal vision. If they have girls, they're all normal vision, but they're also all carriers. Okay, so the answer to that, all normal vision and all carriers. You can try these on your own. Okay, a woman with normal vision who's a carrier. So now her genetics is X to the big N, X to the little n, mates with a normal male. So he's X to the big N, Y. And again, look to see what the phenotype is for sons, and for daughters, tell if they're normal vision carrier, or colorblind. And then a third one, woman with normal vision who is a carrier mates with a colorblind man. Again, give the phenotypes of the sons, and for daughters, normal vision carrier, or colorblind. Okay? Okay, and this is showing the human karyotype, and the kind of thing I would ask you, is that a male or a female? Come on over here, and you can tell that's a female. Now another thing is, chromosomes are usually spread out. It's like if your thread was unwound, and the only time they wind up is right before mitosis or right before meiosis. So whenever you see a karyotype, you'll see that every chromosome consists of sister chromatids and twice the normal amount of DNA. And that's because normally a chromosome would be all spread out and linear. It wouldn't be wound up like this. The only time they wind up is after interphase, when they're getting ready to divide, either in mitosis or meiosis. Okay? So it becomes confusing because people get confused between sister chromatids and homologous pairs. So homologous pairs are one from mom, one from dad. They're the same size and shape. They code for the same genes, but there are thousands of genes, and you're not going to get the same genes from mom and dad. So maybe mom says blue eyes and dad says brown eyes. Mom says make a little bit of growth hormone. Dad said make a lot of growth of hormone. Okay? So those are homologous pairs. Okay, the sister chromatids are, if you kind of draw a line up the middle of the chromosome, it consists of two identical sister chromatids that arose by DNA replication during interphase. So then when the chromosomes wound up after interphase, these two parts of the chromosomes, called chromatids, are identical, and they're held together at the centromere. During meiosis, during crossing over, it's these non-sister chromatids from homologous pairs that will trade places or exchange information. And after they trade places or exchange information, then sister chromatids are no longer identical. Okay? You can see, and if you remember from your karyotype exercise, they're arranged bigger to smaller. Somewhere along the line, somebody figured out that chromosome pair 22 actually has more genes than 21. So trisomy 21, or Down syndrome, happens when you get an extra copy of chromosome number 21. And it just turns out that chromosome number 21 has the smallest number of genes. It might not have less than the Y chromosome, but except for the Y chromosome, it has the smallest number of genes of any of the autosomes. But they'd already put it as 21, so they just kind of kept it there. Okay. Okay, a non-disjunction event. A non-disjunction is a failure during meiosis when the chromosomes don't separate. Either it can happen during meiosis 1, where homologous pair doesn't separate. So here everything starts out right, and then you should have the homologous pair separating. So a big one and a small one on this side, a big one and a small one on this side. Look, here they separate correctly. Okay? But here they stuck together after synapsis. Okay, and so then you wind up over here being short a chromosome. So instead of having 2 chromosomes, one big, one small at the end, you wind up with only 1. You're 1 short, n minus 1. And here they have n plus 1. Instead of having 2 at the end, they each wound up with an extra. Okay? Here everything happens right in meiosis 1. And then meiosis 2 should occur like this. The sister chromatids should separate. And you should wind up with a big chromosome and a small chromosome in the end. So here is normal meiosis. Now here, there was a non-disjunction event during meiosis 2. This pair didn't separate at the centromere like it should. So you wound up, after the second division, with an extra chromosome here, 3 chromosomes, and short a chromosome here. Okay? So this is a young lady with Down syndrome, and she was an actress on the show Glee. And if you looked at her karyotype, you'd see she had 3 copies of chromosome number 21. Now, because people have studied Down syndrome quite a lot, usually it comes from the mother, and usually it comes from meiosis 1. So that's one that we happen to know. It's usually from the egg, and it's usually from meiosis 1. And so you wind up with an extra chromosome, number 21. Okay? That chromosome number 21 would be found in all of her cells. There's nothing wrong with that extra chromosome, but you're getting a triple dose of all the protein products. So it would be like if you're baking a cake, and instead of adding 2 eggs, you add 3 eggs. Instead of adding 2 tablespoons of sugar, you add 3 tablespoons of sugar. So it comes out a little different. And as you get older, there's a relationship between maternal age and the rate of Down syndrome. So after you turn 40, if you're a woman, there's a higher incidence of Down syndrome. There are still more Down syndrome babies born to women of younger age because most people don't wait till they're 40 to have kids. This just gives the analogy I just gave you. There's nothing wrong with any of those genes. It's not a gene mutation, but having that extra chromosome gives you a triple dose of all those protein products. And this is the point I made too, that most trisomies are lethal, and except for one monosomy, all monosomies are lethal. Here's an interesting article if you want to look at it. And this is the fellow who talks about the fact that about 1/3 of miscarriages in the first trimester, if you did the karyotype, you'd find an incorrect number of chromosomes. All right, sex chromosome trisomies. So there are a number of sex chromosome trisomies that are survivable. We'll talk about why that is. If you have a Y chromosome as part of your sex chromosome, you always have the male phenotype. So XYY, that has to come from the sperm. It has to be meiosis II in the sperm. You can think about that. That gives the male. And the Y chromosome trisomy is survivable because the Y chromosome has very few genes. I don't know where these frequency numbers come from, but 1 out of 1,000, that seems, rare, but not as rare as one would think. Down syndrome is something around that as well, and these other trisomies are about that as well. Turner syndrome is less common. I think about 1 in every 10,000, they say. XXY, so again, it has an extra X now, but also a male if you have a Y. And the extra X chromosome is inactivated. We'll talk about that in a bit. And you can kind of see that there are some sort of feminine characteristics like breast enlargement. And then XXX. So now you have an extra X chromosome, and it's a female because it doesn't have a Y. And 2 X chromosomes are inactivated. We'll talk about that in a minute. Okay? Okay, so let's talk about X inactivation in females. So we said in trisomies, it's like making a recipe, and instead of adding 2 eggs, you added 3 eggs. Instead of adding 2 tablespoons of butter, you added 3 tablespoons of butter. And you really don't want to add 3 tablespoons of butter. The recipe calls for 2 tablespoons. So wouldn't it be kind of odd that women have 2 X chromosomes, and that has 1,000 genes on it, that they make twice as much of those recipes as men do? So what happens with the second X chromosome is early in development, one of the X chromosomes is inactivated, and it's random. So in some cells, one X chromosome is inactivated. In some cells, the other X chromosome is inactivated. Of course, men only have 1 X chromosome, so it's always in the active state. Okay? So if a female's heterozygous for a particular gene, she's really a mosaic for that characteristic, with some cells having 1 chromosome and some the other. We'll look at it in cats in a minute. So Murray Barr was a human anatomist, and he discovered this darkly stained object in female cells. And so sometimes it's called a Barr body, named after him. And it's the inactive X chromosome. Okay, so if I take a skin cell from a normal female, any cell except cells from the ovary, and her pattern is XX, then she would have 1 functional X chromosome and 1 Barr body, which represents the normal non-functional X chromosome. Okay, here's an example that will make you understand X inactivation in females. Tortoiseshell cats are black and orange, and they'll have some little white sections as well. Okay? It turns out for fur color, both black and orange are alleles on the X chromosome. Okay? White is a separate gene, and it's not on the sex chromosome. So if you're a cat in embryonic development, one of the 2 X chromosomes is randomly inactivated in every cell. Okay? If you happen to have 2 alleles, if you happen to have a black allele and an orange allele, okay, the black allele gives rise to cells with that gene turned on, and it might lead to a little black patch, whereas the orange allele gives rise to cells that are orange. It might lead to a little orange patch. Okay, the white is a different gene, so the white's there too. And so if you have black and orange, you have to have 2 X chromosomes. So calico cats, not calico, tortoiseshell cats are female. Okay? A male cat could be black and white, orange and white, but not black, orange, and white. All right, and finally, let's to show you there are other possibilities. Here's a chromosomal disorder, and it's actually caused by a chromosomal deletion on the end of chromosome 5. So now it's the opposite. Instead of putting in 2 tablespoons of butter, you're putting in 1 tablespoon of butter. Instead of putting in 2 eggs, you're only putting 1 egg. And you're not missing the whole chromosome. If you're missing the whole chromosome 5, it wouldn't be survivable. Remember, there's only one surviving monosome, having only 1 copy of the X chromosome. But the end of the short arm of chromosome 5 is deleted, and it's serious enough that it causes a serious disorder that's called Karyotype.